Hi there! This is the last video in my series on viruses. Videos 1 through 4 have introduced you to viruses, some of their characteristics, and life cycles. In this video, I'll introduce you to a few more topics. We will very briefly look at how viruses are grown in the laboratory, what a typical virus curve looks like, how to describe virus infections in terms of their duration, and the role of bacteriophages in increasing bacterial virulence. It's a bit of a hodgepodge, but these are the last few topics that I couldn't fit in elsewhere. So let's get started. Your objectives for this video are to briefly describe how viruses are grown in laboratories, to explain how the one-step virus growth curve is different from a bacterial growth curve. You should be able to distinguish between acute viral infections and persistent viral infections. And by the way, you should be able to describe the types of persistent viral infections. You should be able to compare the lytic and lysogenic life cycles for bacteriophages. And finally, you should be able to distinguish between specialized and generalized transduction. There are times when scientists need to grow viruses in a laboratory. For example, when they want to characterize a new virus or manufacture a new vaccine. Whatever the reason, it is essential that they provide appropriate cells for the virus to grow in. 3 common ways that viruses are grown in the lab are shown on this slide. As I mentioned in part 1 or part 2 of this series, if we're interested in studying bacteriophages, those are bacterial viruses, we would have to grow the phages in bacteria. This is done by mixing the virus with bacteria in a test tube or in a container, and then spreading the bacteria out over the surface of the petri dish. The petri dish then gets incubated for a number of hours. After incubation, it may be possible to see small clearings on the plate, kind of like this one or this one. These are called plaques, and each plaque corresponds to one virus that infected one bacterium and then successfully reproduced. Scientists can count plaques to quantify the number of infectious particles in a specimen. Some viruses infect animals and must be grown in animal cells. Embryonated eggs, like this one, are used to grow a number of different viruses for a number of different purposes, like vaccine production, for example. In this case, the viruses are injected into different parts of the embryo in order to grow. For example, herpes simplex viruses can be injected into the yolk sac here, or they can be injected into the chorioallantoic membrane here. Influenza viruses are grown somewhere else completely, in the allantoic cavity around here. Around here. They can also be grown in the amniotic sac here. These viruses can then be extracted and purified to make a vaccination. This is why people with severe egg allergies may be unable to get a flu vaccine injection, that is. A third method of growing viruses is cell culture. In the image on the right side of this slide, we see animal cells growing on the surface of a tissue culture plate, all the way down here. And these cells are growing in a special medium. When the appropriate viruses are injected into the dish, the viruses can begin an infection when they find their receptor on the cells in the plate. These aren't the only methods for growing viruses in the lab, they are just common ones. If we were to follow the growth of viruses in a culture over time, we would notice that its growth curve looks a bit different from that of a bacterium. You'll recall that bacteria use binary fission for growth. You'll recognize the bacterial growth curve here on the right side of the slide. This curve is made by taking samples of bacteria from a culture and then counting the number of cells. Changes in cell number are recorded over time and plotted on a graph. You'll remember that there are 4 phases to a bacterial growth curve. The first phase is the lag phase, depicted here. During the lag phase, bacteria are adjusting to their environment and making changes to their metabolism in preparation for growth, but at this point they're not growing, so our curve shows a flat line as a result. When the bacteria begin to undergo binary fission, they enter the log phase, which you see here. During this phase, the bacteria are growing rapidly, and their growth outpaces the death of bacteria happening in the culture. As a result, we see kind of an exponential increase in number of cells. Remember, they are using binary fission. Eventually, as wastes build up and available nutrients decline, the curve begins to show a plateauing in the number of cells in the culture. This is because the number of bacteria growing is roughly equal to the number of bacteria dying. Then you have the death phase here, where the number of bacteria dying is outpacing the number of bacteria growing. And we see an exponential decline in the number of cells as a result. A viral growth curve, like the one you see on the left here, is called a one-step curve because it is one step. If we were to measure the amount of virus in culture over time, we see that at the beginning of this curve, the number of viruses that we could detect in the culture actually declines initially. During this time, viruses are attaching to host cells and penetrating them. This is followed by the eclipse period, where we would not be able to detect any free, unattached virus in the culture. Basically, what's happening here is viruses are continuing to penetrate the host cell. They are uncoating, doing biosynthesis, and accumulating viral proteins and nucleic acids inside of the host. At the end of this eclipse period, we would see maturation happening, so the viruses would assemble. After the eclipse period, we would start to see the viruses burst forth from the host cells. And they'll burst forth either by budding or by actually rupturing the cell. And this leads to an increase, an exponential increase, in the number of viruses that we can detect within our culture. Now, if this was a culture in the lab, what we would see is that the viral number would basically remain constant, like this. So the curve would actually flatten out like I've drawn here. Inside of a body, like a human body or an animal's body, what we would see is that eventually there'll be a decline, as represented here, a decline in the number of viruses. This would basically signal the point at which the immune system is able to recognize the virus and it starts actually destroying infected cells and destroying the viruses. So the number of viruses goes down. We can describe viral infections in terms of their duration, that is, how long the infection lasts. The first type of infection is called an acute infection. These infections are primary infections of short duration, usually lasting a few weeks or a couple of months at the longest. When we say primary infection, we're really referring to the first time that you come in contact with a particular pathogen and get sick. So these are primary infections that don't last very long. Acute infections are represented by the rapid onset of symptoms, but the infection usually resolves over a few days or weeks, usually because of a robust immune response. Examples of acute infections would be rhinovirus infections that cause the common cold, influenza infections that cause the flu, and norovirus infections, which cause gastrointestinal problems that resolve within 24 to 72 hours. So kind of um, the fact that connects all of these infections is that you do get sick and you feel terrible while you're sick, but you get over these infections within a couple of weeks, maybe 3 weeks at the most. Persistent infections are infections that last for long periods of time, and they occur when an initial primary infection or acute infection is not cleared. Primary infections are those infections that occur again when we first encounter a pathogen. It's the initial cause of an infectious disease. There are three main types of persistent infection. First, you have the chronic infections. These are long-lasting infections. Their duration may be somewhere around 6 months or fewer. Eventually, these infections are cleared by the immune system, though. Chronic infections may be characterized by the continued presence of infectious virus following a primary infection, and they may include prolonged or reoccurring disease. Examples include hepatitis B and Epstein-Barr. So Epstein-Barr is responsible in many cases for mononucleosis. So this is a disease that you may suffer with for several weeks, but eventually you do get over it. Latent infections are ones in which there is an initial acute infection, but the viruses are not completely eliminated by the immune system, and they'll actually go dormant inside of specific cells. What's the implication of this? Well, later, when resistance to infection is lowered because you're under stress, or maybe you're not resting, or maybe you're on immunosuppressant therapies, or you're sick, the virus may become reactivated and infect new cells. A classic example of this is varicella zoster. This is the virus that causes chickenpox and shingles. Chickenpox is a childhood disease that presents with an initial acute infection, characterized by fever, sore throat, fatigue, and a telltale blistery rash. Over a couple of weeks, the blisters dry up and scab, the fever and sore throat go away, and there's a seeming return to good health. Later in life, however, particularly in older adults, the virus may reactivate and become a second, more severe disease called shingles. Shingles is a painful, itchy, blistery rash that appears in stripes along the body. Slow infections are a third type of persistent infection. Here, the primary infection is characterized by an asymptomatic initial disease. There'll be a prolonged incubation period followed by a progressive disease. Rabies is a great example of this. Victims can have this infection for approximately 1 month before showing any symptoms. Once the symptoms begin, however, they become increasingly debilitating. This figure on the right side of your slide is supposed to illustrate the difference between some of these infections. So we have time on the x-axis going from left to right, and number of virions is on the y-axis going from bottom to top. So you can see an acute infection again lasts only a few days. The number of virus particles goes up, let's see, let me do this a little bit better. You have the initial infection where the virus particles increase in numbers. Then the immune system recognizes the virus and fights. This decreases the viral load, the number of viruses around, and that's the end of it. So again, this is like what we see with influenza. For a chronic infection, however, or persistent infection, you'll have this initial acute infection, but the virus doesn't get completely eliminated. And so you may see a long-lasting illness, like you see here, where there's a little bit of virus around causing disease, okay? And the amount of virus can go up and down and reoccur. But eventually, the virus gets eliminated. At the very end over here, you see the latent, an example of the latent type of virus. So here we have an initial acute infection, like this. Then the virus goes into hiding, it's dormant, and we don't see any virus being produced. Then, when we're under stress or immunosuppressed, the virus can reactivate and grow again. So I would be remiss if I finished chapter 13 without talking about the bacteriophages. Bacteriophages are the most ubiquitous, that means the most common entities on the planet. They date back about 3 billion years, and their numbers are truly massive. They're found pretty much anywhere that you find bacteria. We think, or I think it's hard to really estimate how many bacteriophages there are. A couple of things I've seen say that there's something like 10 to the 30 bacteriophages in all of the world's oceans and waters. Another estimate that I saw said there's 10 to the 30 to 10 to the 32 on the planet. So it's hard to even realize how big 10 to the 30 is, but imagine this. 10 to the 30 basically translates to there being about 1 trillion phages. So 1 trillion is a 1 with 14 zeros behind it. So 1 trillion phages for every grain of sand in the world. It's an impossible number to truly wrap your head around. It's huge. In the United States, humans shed about 3 billion coliphages per day. So these are bacteriophages that infect the E. coli in our intestines. 3 billion of them in our bodies, shedding, coming out every day. In about 1 milliliter of unpolluted water, there can be something like 2 times 10 to the 8th phages. Again, another really large number. Phages are found in the air, they're in the food, in our saliva, in the soil, they're in our intestinal tract. They're basically everywhere. So bacteriophages have been really useful to us in science. Some of our early work with them helped us to understand that DNA, not protein or RNA, is the molecule of heredity. Yep, that was scientists working with phages. They've also helped us understand gene regulation in prokaryotic organisms, and they've helped us learn something about virus structure. Believe it or not, at one point, scientists were seriously considering using bacteriophages as therapies against bacterial infections. You might be wondering why we don't use them now. Well, the first phage therapy experiments took place in the 1920s. By the 1940s, this area of study was in decline because scientists didn't really understand phage biology. That was one thing. There was overexuberance or excitement about the potential use of phages for treating bacterial infections, and people got a little careless with their work. And then around the same time, you had the advent of antibiotics, which were easier to handle, and they were very powerful as antibacterials. So we kind of shifted to using antibiotics to treat infections. In some Eastern European countries, however, phages are still in use. And now that we have a problem with antibiotic resistance, there are more and more scientists who are considering going back to using phages as antibacterials. Okay, so it turns out that your bacteriophages go through the same 6 steps that animal viruses that we talked about go through during their life cycle. They have an attachment phase. So a bacteriophage basically attaches to a host cell via protein receptors or carbohydrate receptors. There's penetration. Now, penetration is different for your phages than it was for the other viruses we talked about. Remember with the other viruses, we said that the virus capsid, nucleic acid, everything was brought into the cell by endocytosis or macropinocytosis or by fusion. Well, with a bacteriophage, the capsid and the proteins that surround the nucleic acids of the virus, they actually stay outside. It's just the nucleic acid that gets injected into a bacterial host. After that, you go through the biosynthesis step where we're going to have the production of phage DNA and proteins. Then you'll get maturation where the phage kind of assembles, and then release. And release is a little bit different because we don't really have the budding that we saw when we first started talking about viruses. Instead, the phage causes the bacterium to produce a lysozyme, and as you know, lysozyme breaks down peptidoglycan, so that lyses the cell wall, basically, and the bacterium explodes, releasing new phages into the environment. So there are two types of bacteriophages. You have what are called the lytic or virulent phages. These are phages that are going to lyse the host that they infect, and these are the ones that are most useful as antibacterial therapies. So what basically happens is the phage, the lytic phage, attaches to a bacterial host using these little thread-like structures which are called tail fibers. So they use that to grip onto the host. Then this little stalk region on the phage, which is called the tail fiber, the tail, actually contracts, pushing the tail core into the bacterial cell. Now, there's a lysozyme that's going to help break down the peptidoglycan around this tail so that it can actually penetrate. While that contraction is happening, the DNA genome for the virus gets injected through the tail, through the cell wall, through the plasma membrane, and into the bacterial cell. Once there, the DNA begins to circularize, forming an episome, kind of like a plasmid that's outside of the bacterial chromosome. Now, the cell doesn't know the difference really between the viral DNA and the cellular DNA. So it's going to start transcribing viral proteins, and some of these viral proteins will turn off the ability of the cell to make cellular transcripts and to replicate cellular DNA. Those enzymes that would ordinarily replicate the bacterium, they're going to now start replicating the virus. So we're going to get the production of more viral DNAs inside of the cell. We'll get the production of viral proteins. The proteins will assemble together to form new bacteriophages. Then in the last step of the infectious cycle, the bacterium the bacterium begins to produce a phage lysozyme, which essentially ruptures the cell wall from the inside of the bacterium going outwards. And this releases phage into the environment where it can go on to infect new cells. So that's the first type of life cycle. It's called the lytic cycle, and lytic phages do this. A second type of bacteriophage is called the lysogenic or temperate phage. Unlike the lytic phage on the last slide, this type of virus does not immediately lyse its host cell, and so it's not useful as an antibacterial. The life cycle for this type of phage starts similarly to that of the lytic phages. A bacteriophage attaches to a bacterial host via its tail fibers or pin, connecting with the receptor proteins, lipopolysaccharides, capsules, depends upon the virus, what it attaches to the bacterium through. Phage lysozyme breaks down a portion of the cell wall, and contraction of the phage's tail proteins punches the phage's tail core through the host cell wall and the plasma membrane. At the same time, the phage's DNA is injected into the host cytoplasm. The phage DNA circularizes and gets integrated into the bacterial chromosome via recombination. Repressor proteins from the phage prevent it from entering the lytic cycle, so it rests as part of the cell's chromosome. In this form, the phage is called a prophage. Unlike during the lytic cycle, the phage only replicates when the host replicates, and only one new virus is produced with each replication. The phage can stay like this for years, but when a bacterium is exposed to toxic chemicals or UV light, repair proteins from the bacterium can cause cleavage or breakdown of the phage repressors, and the phage will excise itself from the host and enter the lytic cycle on this side. This process is called lysogenic induction. Phages are not like captains in this way. They don't go down with the ship. They're more like the rats that scurry off and jump off the decks to save their own lives. You may be wondering why a bacterium would put up with having a phage on board. Well, to be honest, if you're a bacterium, being infected with a phage is one of the worst things that can happen. Phages, at some point, lyse their hosts. But lysogeny can benefit a bacterium in the short term for a couple of reasons. Reason number one is that while a bacterium is infected with a bacteriophage, it gains immunity against infection by other phages. It turns out that the repressor protein that holds the phage in the bacterial chromosome also provides immunity. Reason number two is phage conversion. In phage conversions, bacteria pick up new characteristics because they're infected with a phage. The phage could be carrying genes like antibiotic resistance genes or genes that allow the metabolism of unusual substances for energy. These would be genes that benefit a host. The phage could also be carrying genes that would increase the virulence of the bacterium. So here are some examples of bacteria that become more virulent when they're infected with a bacteriophage. Notice that Vibrio cholerae, the bacterium that causes cholera, is on this list. It turns out that when it's carrying a phage, it's able to produce cholera toxin, which is responsible for the horrible diarrhea that accompanies this disease, cholera. Notice that Clostridium botulinum is here. It turns out that without the prophage, Clostridium botulinum is not a deadly bacterium. The gene for making the botulism toxin is on the phage. So when the bacterium is infected with the phage, it can make the botulism toxin and it becomes a deadly bacterium. A third reason that a bacterium may benefit from lysogeny with a bacteriophage is specialized transduction. In this type of transduction, a gene from one bacterial host is transferred to another via a virus. Let's take a look. So remember, we're talking about lysogeny, and during lysogeny, a virus inserts itself, that's the magenta color here, a virus inserts itself into the bacterial chromosome, which is purple. And it happily stays in the bacterial chromosome most of the time. Under stress, or sometimes spontaneously, the virus will actually excise itself from the bacterial chromosome. But occasionally it makes a mistake when it does this. Instead of just picking up viral DNA during excision, the virus may actually pick up viral DNA as well as some adjacent genes that are next to where it inserted in the chromosome. So instead of getting all magenta, you get magenta plus a little bit of purple. In the example here from your textbook, a prophage exists in a galactose-using host containing a gene called GAL. When the virus excises itself from the bacterial chromosome, it takes the GAL gene with it. That DNA gets packaged inside of a capsid, and then when this virus goes on to infect a new host, it injects the viral chromosome, the viral DNA, plus the gal gene that was just adjacent to where that virus inserted in the bacterial chromosome. When the virus integrates into the bacterial chromosome of this new host, the gal gene comes with it. And so this bacterium down here picks up the ability to metabolize galactose because that original gene came from the bacterium up here. So that's specialized transduction. You need to remember that in specialized transduction, specific genes are transferred via a virus. It's not just random fragments like we saw with generalized transduction back in Chapter 8. So this brings us to the end of Chapter 13. In this video, I've briefly described how viruses are grown in laboratories, and I've talked about the one-step virus growth curve and somewhat compared it to the bacterial growth curve. I've also given you definitions for acute viral infections and persistent viral infections. Remember, you need to be able to describe or define the different types of persistent viral infections, so the chronic, the latent, and the slow infection. I've also compared the lytic and lysogenic life cycles for bacteriophages for you, and I've distinguished between specialized and generalized transduction. You might be wondering, what if this material could be on the exam? If I were studying for this material, for this exam, I would probably spend time studying the one-step growth curve and have an idea of what's happening during each of the different phases of that growth curve. So remember we said that the curve, the number of viruses goes down initially. Why is that? And then there comes a point called the eclipse period where you can't detect a virus at all. Why not? Then we saw that the number of viruses in our culture or in our organism go up. What's happening there? And then we saw it go down. So what's happening when it goes down? I described each of those things in this video. If you don't remember them, you might want to go back and look at that again. You also want to remember the definitions for acute viral infection and persistent viral infections. You should also be able to distinguish among chronic, slow, and latent infections because we'll see those again in Chapter 14. For the lytic and lysogenic life cycles of bacteria, I gave you two little tables. I'm mostly interested that you remember the information from those tables. So, for example, under the lytic bacteriophage, one thing that you want to remember is that these phages are going to kill their hosts. They're going to get in, take over their hosts, turn the host into a virus-making factory, and then lyse it. That's different from the lysogenic cycle, where during the lysogenic cycle, the phage actually integrates itself into the bacterial chromosome and doesn't kill it initially. Remember that during a lytic cycle, one phage goes into the bacterium and it can produce tens to hundreds of bacteriophages during that one cycle. During the lysogenic cycle, remember only one bacteriophage is produced with each replication of the bacterial host. Now, should those phages eventually go into the lytic phase doing that lysogenic induction that I talked about, then during that lytic phase, that phage can produce tens to hundreds of phages. You'll also want to remember the benefits that a bacterium derives by allowing lysogeny. So I talked about phage conversion, for example. I talked about immunity, and I talked about specialized transduction. You want to be sure that you can distinguish between specialized and generalized transduction. The biggest difference is in specialized transduction, specific genes get transferred. In generalized transduction, remember, the genome of the bacterial cell gets cut into random fragments, and those get put into capsids and go on to infect new cells. So you get random genes being transferred. So that's the big difference there. I know there was a lot of information in this chapter. I hope you learned a lot, and I hope you've learned things that you can take forward with you into your prospective careers. If you have any questions, please don't hesitate to contact me. I'm more than happy to go through this material again with you if you need it, if you need that. Um, Otherwise, I look forward to starting chapter 14 with you.